Now this one differs slightly from the first one, insofar as this one is better at the 700 wavelength. So we call this one the P700 molecule. And P700 is going to again go and collect all the light, pass it to the special chlorophyll molecules, and those chlorophyll molecules, just like before, are going to give up another electron, just like what happened at this first complex. Now last time when the electron was given up, it was the water molecules that replaced it. But this time, it's not going to be water that replaces it. It's going to be this leftover energy from this electron that just came through the electron transport chain. So it is going to pass through the electron transport chain, and then finally, that last electron is going to be donated right here. In other words, it's going to reduce the primary electron acceptor after the electron acceptor has become oxidized by giving up this first electron. Now, just like over here, this electron is full of energy, and we can use it to do work. That's what the plant is going to do. So there's another protein down here. This one is called ferredoxin, and I will abbreviate it FD. The electron is passed to the ferredoxin, which in turn passes it to another protein over here that we call NADP plus reductase. And the whole job of this enzyme is to reduce NADP+. Remember, reducing is to go and gain an electron. So this electron from the ferredoxin is given to the NADP+ reductase. And what that's going to do is it's going to take my NADP+ and some of this hydrogen that is floating around out here, use that electron, and then use it to make the energy storing molecule NADPH, which, just like ATP, is an energy storage system. It is going to save that energy, and that is also going to be used in the Calvin cycle. So both this ATP and this NADH are going to the Calvin cycle, which we will not be talking about in this particular video clip. But it is the second portion of this cycle where sugars are going to be made using the energy that's stored in the ATP and the NADPH. Now, this second part here that we just talked about with the P700 reaction center and the NADP+ reductase, all of that, this part is what we call Photosystem 1. Remember I told you we would get back to Photosystem 1? There it is. So we discovered Photosystem 2 second. We discovered Photosystem 1, which is this stuff, first. So basically, in order for Photosystem 1 to have happened, you had to have Photosystem 2 happen first, because you cannot replace the electron in the P700 without having that electron left over from the electron transport chain. And you can't have that electron without it having originally come from Photosystem 2. So Photosystem 2 is first, then the electron transport chain, then Photosystem 1. Meanwhile, all those hydrogen that you have been pumping in are now flowing out the ATP synthase through chemiosmosis, and they are causing the phosphorylation of this ADP. Now, because the light energy is what ultimately started all of this, this process of phosphorylating these is what we call photo, because photo means light, photophosphorylation. And I'm running out of space on my board. So that's photophosphorylation. And that is what's happening to my ATP. Now, everything that I've drawn so far, where it goes to this and to this and to this and to this, and heads to the Calvin cycle, that is what we call non-cyclic electron flow. And it's called non-cyclic electron flow because it's not going in any kind of cycle. It's just being passed from one thing to the next thing to the next thing. But there's a catch. In the Calvin cycle, there is going to be a greater requirement of ATP in order to manufacture that sugar then there is a requirement for NADPH. So there has to be a way to produce extra ATP instead of similar amounts. So the way this is going to be accomplished is by a little trick involving the ferredoxin. Sometimes the ferredoxin, instead of passing this electron onto the NADP plus reductase, is instead going to pass it back to the cytochrome complex, which means the energy from that electron is not being used to make NADPH, it's instead being used by the plastocyanin to pump in more hydrogen. More hydrogen inside means a stronger proton motive force, meaning more photophosphorylation of the ATP. So this part, where it does make a cycle, is called cyclic electron flow. So there you have it. That is the basics of the light cycle. Again, photosystem 2 is where this P680 reaction center gets excited by the sunlight, gives up its electron. That missing electron is replaced using an electron from water. The oxygen is released free into the atmosphere. The excited electron moves into the electron transport chain, where its energy is used to do active transport, moving these protons from the stroma into the lumen. And then from there, that electron is then passed on to the P700 reaction center. The P700 reaction center needs that electron because it has recently given up its electron when sunlight excited it. Now, the excited electron from the P700 can be used for one of two things. It can be passed to the ferredoxin, which then uses it to give to the NADP plus reductase, making NADPH. That's photosystem 1. Or it can go through cyclic electron flow and get passed back to the cytochrome, where it's used to pump more hydrogen in to go and make more ATP. All the hydrogen that's in this interior is moving out. That's causing a proton motive force. It's spinning the ATP synthase, which is in turn photophosphorylating this ADP, forming the molecule ATP. End result here, we have oxygen as a waste product released to the atmosphere. We have NADPH and ATP, both of which are energy-storing molecules, headed to the Calvin cycle. I hope you got it.